मिनिमाइज को
मिनिमाइज किया तो बैकग्राउंड में साउंड आएगा ना?
नहीं आया।
उस दिन आया था।
यहाँ पे रिकॉर्डिंग स्टार्ट है ना।
Good afternoon, Chitra. Bhuneshwari ma'am here.
Yes, ma'am. Good afternoon.
Good afternoon, dear participants. It's a very proud moment for us. Today we have with us our own student, Dr. Chitra Rani Rath. She is presently a postdoctoral fellow at Biopharmaceutics Research Institute, Rhodes University, Grahamstown, South Africa. She is her area of specialization is biopharmaceutics. She is presently a postdoctoral fellow, as I told you already. She also worked as a research assistant in Rhodes University from July 2019 to December 2019, as a clinical studies technician from March 2016 to June 2019. She received various awards and scholarships. She was a top performer, one of the top 15% students at Rhodes University by the Golden Key International Honor Society in 2020. She was a gold medalist in M.Pharm Pharmaceutics at Yale University College of Pharmacy in 2014. Also, she was the best outgoing student at Yale University in the year 2014. She got a scholarship and a fellowship for a Biopharmaceutics Research Institute postdoctoral fellowship. Also for a Biopharmaceutics Research Institute Berkeley for doctoral studies at Rhodes University from 2015 to 19. She qualified twice GPAT in the year, once in the year 2013 and once in the year 2014. She received, she awarded the Biopharmaceutics Research Institute travel grant to attend 2 international conferences and 1 training program, that is American Association of Pharmaceutical Scientists Pharma Science 360, San Antonio, US, then Academy of Pharmaceutical Scientists of South Africa, then hands-on training on Hansen vertical diffusion cell system to perform in vitro release testing of dermal products at Dermal Research Institute, Australia. Again, she got various international and national publications. Also, she presented her research paper at various international and national conferences. She is a member of American Association of Pharmaceutical Scientists. Also, she is a registered pharmacist for Goa State. With this brief introduction, I welcome you, Sita, for this session. Over to you.
Thank you very much, ma'am. Is it fine if I stop my video and just start sharing the screen?
No problem.
Okay, thank you, ma'am. Yes. So, good afternoon, everybody. Today I'll be talking about the bioavailability and bioequivalence of topical dermatological drug products. First of all, I would like to thank KLE University's College of Pharmacy and especially Dandegi sir for giving me this opportunity. I was really very nervous because initially I thought it was with students, then I realized it's QIP. I don't know if I'm qualified enough to talk to lecturers yet. So thank you very much. And yeah, so let's start. So what are topical drug products? So topical drug products are the products that are applied directly to the skin or mucous membranes. So when it is directly applied to the skin, it can be because it can be used to treat acne and other skin conditions, or some conditions that have manifestations on the skin like psoriasis, eczema, etc. And so they are meant for local action. So the pharmacological effect of the drug is majorly confined to the surface or the layers of the skin or the mucous membrane. And there are two types of topical products, which is applied to the skin or the surface, external surfaces, and the one is internal, which is applied to the mucous membranes of the eyes, ears, and other cavities. So in case of topical drug products, percutaneous penetration or deposition is not a prerequisite. So we know that it's very often that we confuse transdermal products and topical products and think that they can be tested in a similar way, but this is not true, because transdermal products are administered to achieve systemic drug concentration. So they are absorbed into the bloodstream, unlike topical products, which may be absorbed into the bloodstream, but it is very negligible. So it is less than 1% of the applied drug amount. And for transdermal products, percutaneous absorption is a prerequisite. So if there is no percutaneous absorption in transdermal products, it won't be able to produce its action. But here there is no local drug accumulation. So the methods of BE and BA testing that I will be talking about are not applicable for transdermal products. So now let's talk about generic products. So all of us know that generic products are cheaper, but why are these generic products cheaper than the innovator products? So when an innovator company manufactures a product, it undergoes a lot of research and development. And because of this, there is a lot of cost involved to develop a particular product. So these companies are granted patents, which are for around 15 to 20 years. And when this patent is it is in action, other companies are not free to manufacture the same product. And this is done so that the innovator companies can make some profit and also make up for the expenditure they had because of the development of the product. Okay, so during the 15 to 20 years, generic products cannot be manufactured. But once the patent is expired, generic manufacturers can start manufacturing the generic products. Why are generic products important? So generic products are cheaper. So when the generic products are cheaper, there is market competition, because now the patent has expired and other companies are also allowed to manufacture this product. So there will be market competition. And when there is market competition, the prices will go even lower. And this causes better affordability. So the patients can buy the medications at a lower cost, which can lead to improved patient compliance, because there are certain medications which are more expensive, and then people just weigh the risk and benefit ratio, and then they kind of leads to non-compliance. And when there is improved patient compliance, there is reduced economic burden of public healthcare. So generic products are very closely linked to low costs. Maybe it is at a personal level or at a public level. So now coming to bioavailability, this is the bookish explanation. So for systemically absorbed product, bioavailability is defined as the rate and extent to which the API, the active pharmaceutical ingredient, is absorbed from the drug product and becomes available at the site of drug action. So the rate at which the product is absorbed into the bloodstream, and this is the bioavailability for systemically absorbed products. And for products intended for local action, like the topical products, we cannot actually do this because, like I said, very little amount of the applied medication goes into the skin, so which is less than 1%. And that's why it is actually inappropriate to measure the drug concentration in the blood for topical products. So instead, there are other methods which may reflect the rate and extent to which the active ingredient is released at the site of action. Bioequivalence for systemically absorbed products, it is the absence of any significant difference in the rate and extent or to which the active ingredient is available at the site of action. And it should be, for a product to be bioequivalent, it has to be administered at the same dose and under the same conditions that it is given with respect to the innovator product. So, but for products that are not intended to be absorbed into the bloodstream, it can be assessed by using scientifically valid methods. So what are these scientifically valid methods? I will be talking just after this. So first of all, let's look at the regulations that govern generic products. So when we talk about regulations, the first and the foremost regulatory body that comes to our mind is the US FDA, because they are they are kind of running the market. So this is the history kind of a thing. So the Code of Federal Regulation, Title 21, it has a long chapter which elaborates how the FDA regulates food, drugs, cosmetics, tobacco, and other such products. Then there was a Drug Price and Price Competition and Patent Term Restoration Act in 1984, which was brought into action to control the costs of medication and also to facilitate competition amongst the generic product manufacturers. Then recently, not so recently, but in 2012, there was this Generic Drug User Fee Amendments Act, which was brought into action to speed up the FDA approval. So what happens is when people apply for generic drug approval, most of the times the application is not complete, or it is or there are delays by the FDA. So just to make these processes simpler, FDA brought GDUFA and GDUFA 2 into action. Then recently, this one is very recent, Competitive Drug Generic Therapies FDA draft guidance, which was in 2020, early 2020. So this was brought into account so for the products that don't have adequate generic competition. So there are certain products that are there on the market, just the innovator products whose patents have expired, but nobody has applied for generic applications. So let's see why this happens. This actually happens because when you're applying for a generic product approval, it is quite difficult to apply, and the process is very complicated, which also raises the question, what is the actual use when you are going to invest so much amount? Is it worth to bring a generic product into the market with so much cost? Because the only method currently available is clinical endpoint method, where you have to get a lot of patients, which can run from hundreds to thousands, and it actually costs a lot. So that is why people are shying away. So World Health Organization also has guidelines on the registration of generic products, and it lays down procedures that are required to establish the interchangeability of the generic product with the reference product. Then it is, so the other agency that regulates medicines is the European Medicines Agency. And they also have a draft guideline that lays down the conditions for market authorization of a generic product, and also has legal requirements and procedures that are required to make an application. So once the patent expires, generic manufacturers have to make an application. So sometimes that application is rejected. That is why there is a lot of hesitation from generic product manufacturers. So for registration with the national drug registration bodies, so say somebody is manufacturing a product in India, they will have to show that the rate and extent to which the drug is absorbed from the generic product is equivalent to that which they observed with the innovator product. So that is where comparative bioavailability and bioequivalence studies come into picture. So it is very simple for the products that are absorbed systemically, because they can just do a pharmacokinetic study and then show that the amount, the peak level concentrations of the innovator drug is the same as the generic product. But that is not the case in case of topical products. That's why we need surrogate methods. What are surrogate methods? Surrogate methods are the ones that act, that kind of tell us how the product might have been absorbed and released in the skin. So the bioequivalence of locally acting topical products. So like I said, for B of systemically absorbed products, the methodology and the statistical assessment is well established and has been, people have been conducting these studies throughout. So we have a lot of generic tablets and syrups on the market, but not many creams and ointments yet. So like I said, it is very complicated, daunting, and extremely challenging. So the PK approach is inappropriate because, like I said, it's only less than 1% of the drug that gets absorbed into the systemic circulation from locally acting topical products. And also the amount of the systemic drug concentration does not reflect the concentration of this particular drug at the site of action, which is the skin or the mucous membrane. So FDA has now acknowledged that we need methods that help us to project what will be the rate and extent to which the active ingredient is available at the site of action. So the acceptable methods as of now for topically acting products is a randomized, double-blind, parallel, placebo-controlled study design. So this is a clinical endpoint study where certain people are the patients are recruited. Firstly, these are patients who are suffering from the condition that is being that the product that they have developed is used to treat. So they find the patients, and then some of them are treated with the test drug, some of them are treated with the innovator drug, and some of the people also get placebo drugs. So placebo is the one where there is no drug, sorry. And human skin blanching assay. So this is also known as Vasoconstrictor assay, and it is a very well established method which started in 1995, and our institute has been doing these studies for almost 40 years, and it has been producing very promising results. But it is, the only drawback is that it is restricted to topical corticosteroids. So now, so how do you demonstrate that the generic product is bioequivalent to the innovator product. So firstly, it has to be pharmaceutically equivalent. That is, it should contain the same amount of API in the same kind of dosage form as the reference product. And then it should be bioequivalent to the reference product. So there is another set of guidelines in the Code of Federal Regulations by FDA, which says the generic product should contain the same excipients, should have all the ingredients that are qualitatively and quantitatively equivalent to the reference. RLD is a reference listed drug, which is also the short form for innovator drugs. And then the microstructure, arrangement of matter, and the state of aggregation of the formulation should be similar. So what are Q3? So under Q3, it includes the microstructure, includes the viscosity, in vitro drug release, spreadability, and other such physical parameters. A product is granted a bio waiver if it is Q1, Q2, Q3 equivalent. So it should have the same excipients, it should be qualitatively and quantitatively equivalent to the reference listed drug, and it should also have similar Q3 properties. So this is for systemically absorbed products. It can be demonstrated by comparing the test and reference product using pharmacokinetic parameters, pharmacodynamic parameters, clinical parameters, or in vitro release tests. So first, let's talk about clinical endpoint trials. So these are the most widely used and accepted method of establishing topical bioequivalence at the moment. So all the topical products have to go through clinical endpoint trials unless they have corticosteroids in them. So it has one advantage, that it can directly say whether the generic is as effective or as safe as the reference product. But the problem associated with these trials is that it has low sensitivity and reproducibility. So it depends on the kind of population that we are using in these studies. So this is the problem, because clinical endpoint trials are the only method to demonstrate bioequivalence. But the difficulties we face is ethical concerns, because we have to recruit a large number of patients, and if some patients are given placebo, it could lead to ethical concerns. That is one of the things. They are lengthy and very expensive. There are studies that go on for years, like 2 to 3 years, and sometimes the end results could be inconclusive, and which may lead to inclusion of more participants or termination of the study. So that is why there is a huge regulatory burden, and the manufacturers are hesitant to manufacture any generic product, and it is leading to delayed entry of generics, which is making the prices of the innovator products stay as they were even during the patent, when the patent was valid. So ideally, the prices of the innovator also should go lower once the patent has expired. But the innovator companies take advantage of the situation wherein there is no generic, and they keep on selling it at a higher price. So let's talk about the Vasoconstrictor assay. It is also known as human skin blanching assay. So what is skin blanching? Skin blanching is the pharmacodynamic effect that we see when a topical corticosteroid is applied to the skin. So topical corticosteroid causes vasoconstriction of small blood vessels, which leads to the restricted blood supply to that particular area where the cream or the ointment is applied, leading to lightening of the skin. So let's see how it looks. This is the arm of a healthy study volunteer, and it has been treated with different corticosteroids. So depending on the potency and the strength of the formulation, the intensity of the blanching differs. So like you can see, C, which is clobetasol, is very bright, and hydrocortisone, which is H, is very low. But there is an instrument that we use to measure the intensity of skin lightening. So even if it is not visible to our eyes, the chromometer, we call it a chromometer, it will pick it up. So it is the only well-established surrogate method. Surrogate method is where we don't actually check for the safety and efficacy of the product, and then we just correlate the pharmacodynamic approach. The pharmacodynamic effect that we observe with skin blanching is correlated to its absorption or permeation into the skin. So the intensity of, like I said, the intensity of the skin lightening or blanching is proportional to its potency. Skin blanching was initially recorded by Hollander et al. In 1950, and it took a very long time, 12 years, for McKenzie and Stoughton to use it for bioequivalence and bioavailability and bioequivalence assessment. So until recently, until 1995, there were operators who were trained to look at these skin blanching and rate them on a scale of 0 to 4 to 5, depending on the skin intensity, the intensity of lightening. But this was very objective, and then it depended on who was looking at it. So it varied greatly between different observers. And that's why it was replaced by an instrumental method in 1995. So this is the guidance. Topical dermatologic corticosteroids in vivo bioequivalence was a guidance, and we are still following that guidance to do the bioequivalence studies for topical corticosteroids. So this is the machine that we use. It is called the chromometer. So the one that looks like an arm is placed on the site, and then it kind of takes a picture. So it flashes a white light. So it is portable, and it uses triced stimulus colorimetry, which means it uses 3 different wavelengths and it emits white light. So it exactly looks like a flash. So it flashes on the application site and then measures the intensity of the reflected light through 3 wavelengths. So these signals are converted into 3 coordinates. So one is luminosity, the second one is amount of green or red, the third is amount of yellow or blue. For this particular study, we use the A scale reading. So A scale reading measures the amount of green or red. So when there is no medication applied at baseline reading, there will be normal blood flow. So it will measure the redness. So say the redness is, the number is 9. And when you apply the topical formulation and then check it after it blanches, that number 9 will go to say 7 or 8. So that difference tells us that the product is acting and how much has gone in. So this is A scale reading is our point of interest. So these studies are divided into two parts. One is the pilot study and the pivotal study. So pilot study is done to determine the dose duration for which the product has to be left on to obtain proper response. Okay. So this pilot study is done only using the reference product or the reference listed drug, because we know that the reference listed drug is already on the market and it is doing what it is supposed to do. And the readings are taken for 24 hours, and the A scale reading, whatever the A scale reading we get, it is plotted against the time, and then we get an area under the curve. So we use Emax model for the analysis, and this is the Emax model equation. So it tells us, if you can see ED50 here, that is the time at which half the maximum response is obtained. So the maximum response is the maximum AUC, which is the Emax. So the maximum AUC is the maximum response, and the half maximum response will be Emax divided by 2, and the time that corresponds to Emax divided by 2 is ED50. So ED50 is the time which is used to compare the test and the reference product. So, but there is one more consideration here. We have to find out whether the data from that particular person is evaluable or not. So for that, we have to use this equation where the AUEC at D2. D2 is ED50 times 2, and D1 is ED50 times half, ED50 divided by 2. So it just says that the volunteer is showing dose dependent or dose duration dependent response. This is essential because if the ratio is less than 1.25, the person might show positive, false positive or false negative effects. So that is why we use the data only from the people who have over the ratio over 1.25. So say we take in 60 participants, there will be only about 40 participants whose data is evaluable. So, and then there is pivotal study. This is a study where the generic drug, which is the test drug, is compared against the reference drug. So this is where we take the ED50 that we get from the pilot study, and the AUECs are calculated, and a confidence interval is built. So the AUEC of the test by the reference should lie between 0.8 to 1.25. So if it is less than 0.8, it means that that it is not bioequivalent, and even if it is more than 1.25, it is not bioequivalent. So the limitations of this method is that only light-skinned individuals are used in this method. So I call this study very racist because it is only mostly Caucasian people that can be recruited for this study. So it has high variability. That's what I said. It has to be more, the AUC ratio should be more than 1.25. That's why to get 40 evaluable data points, you have to have at least 60 participants. So there are other promising approaches. One is the dermatopharmacokinetic method, which is tape stripping. It is a very simple method, but can be very time consuming. Dermal microdialysis, this requires a lot of expertise. Then dermal open flow microperfusion, which is very similar to dermal microdialysis, but uses a different kind of semi-permeable membrane. I will be going through this in detail in the later slides. And then in vitro release testing. So first coming to tape stripping, this was one of my this was a part of my thesis, so it is very close to my heart. So this is relatively non-invasive. If I tell you, it is a very simple method. So we just take any book tape and first test if it is not irritating to the skin of the subjects, and if it is irritating, then we just change the tape. But usually we use medical tape or normal sellotape. So you just apply the tape and then remove the layers of the skin. So the layers of the skin that you remove contains stratum corneum. So this stratum corneum will contain some amount of the drug that has gone through it, and when we extract it and analyze it on HPLC, it will show a peak. So let's see what it is about. So it is like pharmacokinetics, but dermatopharmacokinetics. So the major difference between pharmacokinetics and dermatopharmacokinetics is pharmacokinetics will have a concentration as a Cmax, but here we have Qmax, which is a quantity. So it will be microgram per centimeter square or milligram per centimeter square or nanogram per centimeter square. It is never milligrams, it is either micrograms or nanograms. So the concentration of the drug in the stratum corneum indicates the topical bioavailability of that particular product. The only issue with this is it doesn't have any universally accepted protocols. So in 1998, FDA tried to set down the procedures for tape stripping to be used as a method for bioequivalence assessment. But in 2002, they had to withdraw this guidance because there were two companies who were working on the same drug which produced contradictory results. So because of this particular guidance was withdrawn and never visited back. But there are different study groups, different groups in the world that are working on this, and they're trying to optimize the conditions so that there is less variability. This tape stripping study also involves a pilot study and a pivotal study. So the pilot study is basically to make sure that the method that you're using is optimized and is giving out, and then you make sure that it is quantifiable, and I will just go through it now. And then there is a pivotal study which is used for comparative BE assessment. So in pivotal study, all the participants get one treatment site with the reference and one with the test. So that is direct. We compare the two formulations directly on the same participants. So this is the tape stripping procedure in short. So first is template application. If you can see the white color thing, it is just a tape which has a square drawn on to it. This is done so that the application area is restricted. And the second thing is we also have to restrict the area of stripping. So it is necessary to cut the tapes of the same size so that the stratum corneum from surrounding areas is not taken out. Then second one is product application. So in that white window, the window that you can see, that is where we apply the product. And the product is applied and left on for a certain number of hours. So first for the pilot study, what I did was I left it on for different time points and then removed it after those time points and stripped the skin. So then we place the tape strip, then apply uniform pressure using a roller or you can use a roller, you can use a thumb or a spatula. I used a roller because I thought that rollers are more make it more uniform. Then the tape is stripped using a single upward pull. And this tape stripping is not done only with one tape strip, it is done with multiple tape strips. So we usually need 10 to 20 tape strips to remove almost 80% of the skin. So after that, these tape strips are weighed. So the tape strips are weighed before the tape stripping procedure and after the tape stripping procedure. So when we do this, it gives us the amount of stratum corneum that is removed. And for weighing, we need a very sensitive instrument. The one that I was using was very sensitive even to air, just normal air. So we were using an anti-static device to remove the static from the tapes and then weighing it. The next part is extraction and analysis. So these tapes are then rolled and put into vials, and then some, depending on the solubility of the drug, we use the solvent and then extract the drug out of it and then analyze it using UPLC or HPLC. So because we are using such a large number of participants, we cannot say that the skin properties in different people, if in different individuals is similar. So there is a need to standardize the skin thickness. So this is done using TWL measurements. So TWL means transepidermal water loss. So when we remove a layer of the skin, there is some water that is lost, and that is what is converted. I will show you just now. So that is what is converted into skin thickness. So this is 1 by TWL, and it is plotted against the skin thickness. So the amount of skin removed is converted into micrometers, and then it is plotted. So the point where the points on the y-axis meet the x-axis, it is the skin thickness. Okay. So after the product is removed and analyzed, after the drug is stripped from the skin and analyzed, it is plotted against the time. So it gives a dose, it is kind of a dose response curve, and it, yeah. So this is the kind of dose response curve. So it can be, here I have plotted it against the fraction of stratum corneum removed. So it was around 62 to 63% of stratum corneum that was removed, which was plotted against the amount of drug per tape strip. So I used 20 tape strips, so there are 20 points here. And this, the area under this particular curve is used, the area under this particular curve is used as a measure of the extent of absorption. So Qmax is the maximum, the amount of drug that is the maximum amount of drug that is permeated through the skin, basically. And like I said, TWL measurements are used to standardize the stratum corneum thickness. And this is done only on the blank side. So TWL thickness is not done on the site that is that has received any kind of treatment. So it is done on the blank side, and then it helps to determine the stratum corneum thickness per patient. So this is the instrument that we use to determine the transepidermal water loss. So you can see the round part here that is placed on the arm, and soon it is placed on the arm soon after stripping one tape. So we have to do this. Sorry. Yeah, we have to do this after stripping each layer of the skin. Okay. This I did. Yeah. So the problem with tape stripping is it is very variable. So there is high variability associated with this method. So this is what I did. First was tape selection based on the adhesive sensitivity and irritation and recovery. So recovery is one of the major parts. Before selecting a particular tape, we have to make sure that it doesn't interfere with the HPLC analysis of the drug. So it is one of the major parts, tape selection. The second is determination of dose duration. So this is done after the pilot study. And the third is number of tapes. So the number of tapes differs with the type of tape used. Some tapes that stick very firmly to the skin might remove 80% of the stratum corneum with 10 tapes, and some that are not very adhesive to the skin will remove will need like 20 tapes to remove 80% of the skin. So the tape that I used was a Scotch tape, and I needed 20 tapes to remove at least 70% of this stratum corneum. The dose applied, so we have to be very careful about the dose that we apply as well. And the gravimetric analysis. So this is one of the major concerns, and it is one of the most time consuming parts of the study. So first we have to cut the tapes because it is a normal tape that we use for our books and binding and gift wrapping. So we have to cut it 2 by 2, just over 2 by 2 centimeter, and it is like 20 tapes per side, and there are sometimes 10 sites. For 10 sites we need 200 tapes, so we have to individually cut these tapes and weigh them before doing the tape stripping and after doing the tape stripping. So it is one of the most important, most crucial parts of the tape stripping study. So the dose duration study, this particular study also uses the Emax model where we determine the ED50. So this is done using a reference product. So this particular study is a case study where the reference product was Metro Cream, which contains 0.75% of metronidazole. It was done on 6 healthy subjects, and these different dose durations were used. So 1 hour, 2 hours, 4, 6, 8, and 10 hours. And it was done on the left arm of all the participants, and one dose duration was assigned to one application site, and there was one blank site. So this is how the arm looks. So A, B, C, D, E, F, and the blank. So the blank was kept uniform for all the participants, and A, B, C, D, E, F was randomized between the participants. So for this study, I got an Emax of 85.29. Like you can see, it is this particular point, 85.29, and the ED50 is half of that. So half of that will be somewhere here. So it is 60.89 minutes. Since it was 60.89 minutes, it was a win-win situation for me because then I could use 1 hour as the dose duration for pivotal study. So in the pivotal study, I did bioequivalence testing as well as confirmed bioinequivalence. So sometimes there can be false positives or false negatives. To rule out these, we have to include some products that we know for sure will be positive and some products for sure which will be negative. So let's see how it was done. So I had 10 healthy human subjects, and we used both the arms for this study with 4 application sites and 1 blank site on each arm. So the reference product was the same. Test products, there were 3 test products. So one test product is the first T1 is the one that has the same strength as the Metro cream, which is the reference. Then one that has 25% less strength compared to the reference. And then another one that has 26% more strength compared to the reference. So as a part of positive control, the reference was compared to itself, because we know for sure this has to be bioequivalent. So we compared the reference to itself, and the negative controls that contained less and more amount of cream compared to more amount of the drug compared to the reference were used as negative controls. So if at all the positive control comes out to be non-bioequivalent, then it is a failure. And if at all the negative controls come out to be bioequivalent, it is also a failure. So the results from the bioequivalence study are here. So like you can see, the T1 versus R, it is kind of ideal. It was 1.007 and it was in the confidence interval of 0.8 to 1.25. And this one was where the reference was compared to itself. It was 1.05 and the confidence limits were also within the specified interval. And this is the one where it was the T2 contained lesser amount of metronidazole compared to the reference. So you can see it fell outside the confidence limits. Although it did not fall completely outside the confidence limits, it did fall slight. So it should be less than 0.8 also. It is, even if one limit is outside the confidence interval, is outside the specified limits, it is non-bioequivalent. Similarly for this one as well, it was 1.25 and it is 1.18 to 1.33, which also means this 1.33 is outside the confidence interval limits of 0.8 to 1.25. So this was also non-bioequivalent. So now coming to dermal microdialysis. So what is dermal microdialysis? So this was initially used for PK studies, for normal PK studies in humans. And then only for the past few years, it has been used for dermal microdialysis. So there is a probe, like you can see this probe. The blue part here is the probe, which is placed just under which is placed in the epidermis, and sorry, this part is the probe which is in the dermis, and it is made out of a ultra thin semi-permeable fiber structure. So this semi-permeable fiber allows the topical formulation that is applied on top to pass through it. So there is a continuous flow of perfusate from a pump, and when it goes through the semi-permeable membrane, it also carries the drug molecules that have been absorbed, and then we can analyze it using HPLC or UPLC. So yeah, it is a sterile buffer that is used, which mimics the physiological fluid. So the gray area here, the gray part here, which looks like a syringe, is a microdialysis pump, and it pumps the fluid at a very low rate, which is between 0.25 microliter per minute. So this, even though it is invasive, it is not painful. This probe allows bidirectional exchange of diffusible molecules, which means some of the fluid might go into the the deeper layers of the skin, and some of the molecules that are in the dermis come into the semi-permeable membrane and the fluid that gets collected here. So it is also one of the benefits as well as limitations of this, because then it has to have sufficient recovery for this to be a method that can be used with full confidence. So the applications, it is used to quantify the endogenous substances and absorption of exogenous substances. So exogenous substances are the ones that we apply from outside. Endogenous substances are the ones that are produced by the body. So it is also used to assess drug permeation in healthy as well as damaged skin. For dermatopharmacokinetics or tape stripping, we cannot use we cannot use damaged skin, but this can also be used in damaged skin. And this is a major advantage, because then we do not have to extrapolate the results from healthy volunteers to the patients. This can directly be done in the patients. It is also used to determine bioequivalence of certain topical formulations. And yeah, like I said, it is invasive, but it gives detailed pharmacokinetic data. So this is as good as getting a profile that we get for the substances that are absorbed into the bloodstream. Yeah. So simultaneous assessment, this can be done. There can be various probes that are attached on the forearms or thighs of a person, and many different formulations can be assessed at the same time. So this is a major advantage, because the test and the reference can be simultaneously assessed. Major concerns about this are, it is difficult to select a perfusate that is tissue compatible as well as drug soluble. This problem can be overcome by using a tissue perfusate that is a lipid emulsion, because then it will help solubilizing the lipophilic substances as well as lipophilic drugs as well. So it has a very low recovery for lipophilic or highly protein bound drugs. So that is one of the major concern. And probe calibration is required. So we have to make sure that the probe is properly calibrated, because otherwise it will give rise to a high degree of variability. And the perfusion rate and the sampling have to be optimized, and also the operators conducting these studies have to be well trained so that they do not hurt the study participants. Yeah. So this is one picture that I took when I went to Joanneum Research, and like you can see, she has 6 probes on her thigh and 6 probes on her arms. So it is like duplicate. It has 3 formulations and they are assessed in duplicate. This is one of the studies that was done at BRI by one of the students in 2008, and he used 4 probes and 4 application sites. So it was 4 different applications, and which were 1.5 meters apart, and this was used for ketoprofen analysis. And this is the profile that you get after analyzing the dialysate concentration and plotting it against the time. Because this gives time dependent response, it is one of the most preferred methods, but there are so many difficulties that this is hardly being used. Then coming to open flow microperfusion. Open flow microperfusion is a relatively new method. It was only launched in 2011 for it was used for brain initially in animals, but it was used for dermal bioequivalence and bioavailability only in 2011. So the major difference, the principal difference between the dermal microdialysis and open flow microperfusion is the probe design. So the probe design varies majorly because the OFM probe is an open exchange area. Like you can see, it is just a porous probe which has which is not like the semi-permeable membrane. So since the probe is open, it exchanges the fluids very the exchange of fluids is very simple. There is this push-pull pump that is used to to push the perfusate in and then pull the resultant out. So this this overcomes the limitations associated with dermal microdialysis because micromolecular size and protein binding won't be an issue in this case. The application, it helps to establish a relationship between the amount of drug in the dermis and its pharmacological response by simultaneously measuring the drug product and a pharmacodynamic marker. So topical bioequivalence, where the skin barrier is perturbed, like in case of patients, it can be used to check how the bioavailability of the drug has been altered by that particular condition. This one does not involve the use of any membrane and instead uses a push and pull pump. So these are the push and pull pump. The one that is marked orange pushes the perfusate in, and the one that is marked blue pulls the air out. So those methods were all using participants, but this is in vitro release testing, which does not use any participants and can be done in the lab. So like we know, for most extravascularly administered solid dosage form, particularly the oral dosage forms, there is dissolution testing, which can be used to determine drug release. This has been well established, and we've been doing it from our undergraduate days. But there is no standardized test applicable for all semi-solid dosage forms. They have been trying to do this, but there have been difficulties for this. So FDA has one guidance called Scale Up and Post-Approval Changes Semi-Solid Dosage Forms Guidance. So it lays out the procedures that a particular company may follow if there are minor changes done to a particular formulation. There is a compendial method in the USP, which is laid down in the chapter 1724, and it describes the different types of vertical diffusion cells that can be used to assess in vitro drug release. The other applications of this is to support formulation development, to compare generics with innovator formulations, to characterize release data from formulations used in clinical trials. And there are some FDA product specific guidances. So these guidances lay down the method, the procedures for in vitro release testing, but only for a specific product. So there are draft guidances for acyclovir cream, acyclovir ointment, but these methods cannot be extended to other drug products. So that's why they are called product specific. And they, in vitro release testing, in combination with other studies like clinical endpoint studies or in vitro permeation studies, can be used to apply for a biowaiver. So biowaiver is where you don't have to do the actual studies in the patients. This is one of the cells that are used, vertical diffusion cells that are used for in vitro release testing studies. So this is manufactured by Permagear. Like you can see, there is a donor compartment, donor chamber on top. There is a membrane in between, and then there is a France diffusion cell. So it looks exactly like the France diffusion cell. The only difference is there is a heat jacket through which the water is circulated, and I'll show you. This kind of an assembly is used where multiple formulations, multiple samples can be tested at the same time. So this is a 6-cell manual vertical diffusion cell system. So before starting an IVRT method, we have to make sure that the particular instrument that we are using is capable of measuring the drug concentration appropriately. So first, we have to make sure the capacity of the cells is perfect, the diameter of the orifice is well within the range, the temperature of the receptor medium should be decided. It is usually 32 degrees Celsius if specified otherwise for topical drug products. The speed of the magnetic stirrer, so depending on what speed we set the magnetic stirrer on, it might increase or decrease the release of the medication from the dosage form. And the dispense sampling volume, the amount of volume that we withdraw for sampling also needs to be analyzed, and we need to make sure that the amount that we are withdrawing is consistent throughout the study. And benchtop levelness, this is benchtop levelness, no direct exposure to sunlight, and failure resistant power supply are the environmental factors that we have to consider. So when we develop an IVRT method, we have to make sure the receptor fluid is good enough to solubilize a proper amount of the drug in itself. So what is the proper amount of drugs? So we know, say a product contains 0.62 milligrams of the drug. So we have to make sure that the receptor fluid is capable of dissolving 10 times 0.62 milligrams of that particular drug. So for that, we have to shortlist a few receptor fluids that we feel might be compatible, and then do the studies and do the solubility studies and choose one particular receptor fluid. The second one is membrane screening. We have to make sure that the membrane that we are using is not causing any of the drug to get stuck in it or bind to it, which might lead to lower concentration of the drug in the receptor fluid. And then the stirring speed. The stirring speed should be optimum. It is usually 600 RPM. And then the sampling duration. So sampling duration is usually 6 hours, but sometimes there is a kink that we observe in the profile. And for IVRT, it is necessary for the profile that we plot, which is, I'll show you. This is the profile that we plot. So the cumulative amount of drug released versus the square root of time. So the regression coefficient for this line should be greater than 0.9. So if at all there is a kink in if at all this line starts bending, and if at all the R square value is less than 9%, we have to find out the part where the R square value remains consistent and is more than 0.9%. So after these things are done, we have to go for method validation. So before comparing the reference with the test product, we have to validate the method and make sure that it is doing what it is supposed to do, like we do for other methods also. So we have to make sure that it is linear, precise, and reproducible. So this is an example where the 3 runs were done and then made sure that it is linear, precise, and reproducible over the 3 runs. Then sensitivity, specificity, and selectivity. So this is very important because we need to make sure that the method is sensitive to the amount of drug that is coming out, specific to that particular drug, and is selective as well. So this is one of the plots that I have done. So here it makes sure that different concentrations, different product strengths have different release rates. So the release rates of the different the runs using different product strengths should also be a straight line. And here we got 0.9994. So the next part is robustness. In the part where we do robustness, we tweak the conditions a little bit. So say increase the stirrer speed or increase the heat of the increase the temperature of the fluid. So for robustness, it should be less than 15% of difference between the one that we have optimized and the one that has been used after tweaking the stirring rate and the temperature. Then recovery, need to make sure the recovery, we recover less than 30% of the API. This is a weird thing because in other cases we want to make sure as much we want to make sure that most of the drug is released, but in this case the recovery should be less than 30%, because once it is more than 30%, the profile starts developing a curvature and then it is not consistent. So when it starts developing a curvature, it is more than 0, it is less than 0.9. So the regression coefficient is less than 0.9, which makes the line not linear enough. Then the sameness assessment. So for IVRT, we do not call bioequivalence assessment because it is not done in humans. So it is called sameness assessment. So used a test product which contains 0.75% of metronidazole versus the reference cream, which also contains 0.75% of metronidazole. So when we run, we have to make sure the reference covers all the 6 compartments on the vertical diffusion cell system, and also the test covers all the 6 spots. This is because we have to rule out that the results might be because of the placement of the formulation. So this is the result from the sameness assessment. So both the test and the reference were found to be equivalent. Then assessment of discriminatory power. So just like tape stripping, where we tested for false positives and false negatives, this one also was used to, we also used IVRT to rule out false positives and false negatives. So the same negative controls were used against the reference. And since there were 3 formulations, we had to do 3 different runs so that all the products got all the slots on the system. So this one is when T2, which contains less metronidazole compared to the reference, was compared to the reference. Like you see, it is slightly less than the reference. And then this is where the test product, which contained more metronidazole than the reference, was compared to the reference. And here also the test was slightly higher as expected. So this rules out the possibility of false positive. Then, so now the concluding remarks. So we've seen that there are different methods, but none of these methods are standardized. So there is an urgent need to develop a more economical alternative for bioequivalence assessment of topical products, especially because then it will help to increase the increase the speed at which this generics will enter the market. So it is noted that the price of the medication goes significantly low once the second and the third generic come into the market. So it is a very important thing that there are methods that help us to bring more generics into the market. Then the next is refinement and optimization of existing strategies. Like we said, there is tape stripping, microdialysis, microperfusion, and IVRT, but these methods don't have any standardized operating procedures, no SOPs, no standard procedures. So everybody is doing according to their own convenience. Because of this, different labs get different results, and none of this method is ending up in any of the guidances. So there are just draft guidances, but there are no proper guidances. So that's why we need standardized protocols and procedures for these surrogate methods. Okay, so I would like to thank KLE University's College of Pharmacy, especially Dandagi sir and Patnala sir, because of whom I am at Rhodes. So I would also like to thank Biopharmaceutics Research Institute and my colleagues here and my supervisor, Prof. Isadore Canfer. Thank you very much. And do let me know if you have any questions.
Thank you, Sitra. Dear participants, now the session is open for questions. We have one question in the chat box.
What is the approximate cost of this chromometer?
So chromometer is 1 million rand. So it is around I think it is around 1 million rands.
Oh.
So which is around 40 lakhs.
Oh. Next question.
Let me cross check.
Oh.
Okay, so it is $9,999.
I have another query also along with that. If we are using any topical formulation, maybe for a cosmetic rather than having an active ingredient, if we want to check any moisture content present in the skin, whether this chromometer will help us, is it something like the Teva meter what is being used? Whether it will help in that or it will be only for the color differentiation? So
chromometer is only used for color changes, but like you said, for cosmetics, you can use the TWM method, the tapometer.
Yes, yes. Thank you.
Any other questions? Is there any questions in the chat box?
No.
Okay. Any other delegate wants to ask any other question? Oh, if there are no questions, we will proceed. Yeah, it was a very informative session, Sitra. Sitra gave overview of VAB studies of topical drug formulations, all her experiences she shared, which helped our students as well as the delegates. It was very informative session with all elaborative explanations and all. Once again, on behalf of ALS College of Pharmacy and my own behalf, I thank you very much, Sipra, for accepting our invitation. In future also, we will expect more and more presentations from you. Thank you once again.
Thank you so much, ma'am.
Yeah, dear delegates, with this we have come to the end of today's program. So see you all tomorrow morning with another interesting session. Thank you one and all. Thank you. Thank you, Sipra.
Great.